Hey everyone, welcome to our lesson on the connective tissue physiology. This includes the bone and joint and the integumentary system. I am Ophelia Ibanez and I will be going through this topic with all of you guys. So this is the outline for today. We are going to discuss your connective tissue and then a little bit on the bones, mostly on the bones and joint, and finally your integumentary system. These are our learning objectives for today's lesson. You can read through them, pause so you know what to focus on at the end of the topic, and the list of the entire learning objectives will be found in your student guide. These are our topic outline for the bone and joint, and then we will also have a topic outline for our integumentary system. We will be discussing a lot more on the connective tissues and bones rather than the joint. Our integumentary system is a bit more lengthy than our bone and joint or our connective tissue part, so please be wary of that. Take time to pause and digest the topic before proceeding. So let's start off with our connective tissues. So your connective tissues are aptly named. They connect epithelia to underlying body structures, they link muscles to bones, and hold joints together. However, your connective tissues provide the body with more than structural connections and form. They also play vital roles in defense, repair, storage, and nutrition. The extent and composition of the extracellular matrix, or our ECM, is a key feature of connective tissues. Unlike epithelia muscle and nerve tissue, which consists mostly of cells, connective tissues contain significant amounts of extracellular material, which may be synthesized by only a small number of resident cells. Let's now talk about the functions of the connective tissues. First and foremost, it has a role with structural support, where the connective tissues form the structural framework on which the rest of the body is assembled. It also plays a role in nutrition, where epithelia are not directly vascularized, whereas arteries and veins supplying organs are surrounded by connective tissue. So the exchange of nutrients and waste products between the blood and capillaries and venules and cells of an organ occurs by diffusion through the connective tissues. It also plays a role with storage. So ECM of the connective tissue is an important site for storing water. It also stores electrolytes like sodium chloride and plasma proteins, and lipids are stored by adipocytes. Additionally, our ECM provides a physical barrier against microorganisms that manage to penetrate epithelia in the basal lamina, which helps confine infections by these invaders. Thus, connective tissue is often the site of inflammatory and immune responses to pathogens. These responses are first mediated by connective tissue residents, principally macrophages and mast cells, and by transient cells, which may be recruited in large numbers from the blood in response to signals generated by an acute infection. And finally, or another one, is its roles in repair. So tissue loss due to infection or injury usually stimulates the growth of local connective tissues in an organ. The deposition of new ECM can help close wounds and provides a support for organized tissue replacement. In some cases, total repair is not possible, and connective tissue scars fill in gaps created by lost parenchymal tissue. Persistent damage to an organ may cause excess production of connective tissue, resulting in diminished function of the remaining parenchymal tissue. This condition is known as fibrosis. And finally, it plays a role with information. So many molecules secreted by cells that are involved in cell-to-cell signaling, for example, growth factors and inflammatory mediators, do not diffuse freely through connective tissues but are bound by ECM components. This binding can result in several effects, including limiting the spread or producing locally high concentrations of the signal. Let's now talk about the cells of connective tissues. These are the permanent cellular residents of connective tissue. So we're going to start off with fibroblasts. Fibroblasts are typically the most abundant cells found in connective tissues and have the following properties. So they synthesize and secrete fibers and ground substance, which is the major component, or these are the major components of ECM. They are normally non-mitotic. However, during tissue repair, fibroblasts can proliferate rapidly while also increasing their synthetic activity. They also contain abundant rough endoplasmic reticulum and an expanded Golgi apparatus when actively synthesizing and secreting matrix components. Fibroblasts can also produce and export factors that influence the growth and differentiation of neighboring cells. And then next we have our adipocytes. Our adipose tissue is a special connective tissue, and this is discussed later in this chapter. Adipocytes are also found scattered in other organs and have the following properties. In adults, adipocytes store lipid in a single droplet that occupies most of the cell's volume. The lipid is extracted during the processing required to prepare a slide, leaving a large, clear center surrounded by a thin rim of cytoplasm containing a flattened nucleus. The number of adipocytes is determined early in life, and these cells rarely divide in adults. Adipocytes produce a variety of endocrine hormones and cytokines or cytokines involved in regulating metabolism and the immune system. Alright, next we have macrophages. They have several functions as follows. They phagocytose and attempt to destroy foreign particles, microorganisms, and damaged tissues. They are also involved in the normal turnover of ECM by secreting hydrolytic enzymes, such as collagenase. This activity is increased during inflammation and repair. Macrophages are also key players in immune reactions. They can initiate immune responses by presenting antigens to lymphocytes and by secreting cytokines, which are the signaling molecules that are important for immune system activities. Next, we also have mast cells. Mast cells resemble basophils or basophils found in the blood. They also are derived from bone marrow precursors and function as follows. Mast cells store chemical mediators of the inflammatory response in large basophilic granules. They are responsible for allergic reactions. So, mast cells are responsible for allergic reactions, also referred to as immediate hypersensitivity reactions, which occur when these cells rapidly release granules when exposed to an antigen. Chemical mediators stored in granules include histamine, which promotes vascular leakiness and smooth muscle contraction. We have heparin that blocks local coagulation, and factors such as eosinophil chemotactic factor, which attract other immune system cells. As mentioned previously, some cells in connective tissue are permanent residents. For example, fibroblasts, as discussed in the previous slide. However, some connective tissue cells are transients and enter the tissue from small blood vessels via diapedesis. This entry is often in response to local signals, such as those generated by microbial infection. Transient cells include plasma cells, lymphocytes, neutrophils, and eosinophils. Let's start off with plasma cells. Plasma cells may be found in any connective tissue and are common in the lymph nodes, spleen, and mucosa of the digestive system. The average lifespan of a plasma cell is 10 to 20 days. With leukocytes, we have three types. We have your lymphocytes, your neutrophils, and your eosinophils. Let's start off with lymphocytes. So again, leukocytes, they are, besides macrophages and plasma cells, these are types of white blood cells as well. So lymphocytes accumulate at any site of acute infection and tissue inflammation and are commonly found in the loose connective tissue, or your lamina propria, of your respiratory and gastrointestinal tracts. In these areas, many lymphocytes may be long-term residents. In other sites, they may pass through quickly. Another type of your leukocyte is your neutrophils. These cells are common in blood but are rare in normal connective tissue. Neutrophils are attracted in large numbers to sites of bacterial infection. Neutrophils contain membrane-bound cytoplasmic granules that contain a variety of substances, including defensins, which can kill bacteria. So, when attracted to the site of a bacterial infection by inflammatory signals, they avidly phagocytose bacteria and destroy them. Another type of our leukocytes is our eosinophils. These cells also are rarely found in normal connective tissues but may be recruited from the blood in large numbers by signals generated from allergic reactions and parasitic infections. Eosinophils can kill parasites by releasing cytotoxins from cytoplasmic granules. They also release enzymes that destroy histamine and heparin and thereby have the potential to modulate allergic reactions. So we're now going to talk about the skeletal system, so more on the bones now. Let's start off with the extracellular matrix. The main components of the extracellular matrix are our fibers and ground substance. We have our collagen fibers. Our collagen fibers, they assemble or they have assemblies in bones. We have our osteoblasts that produce two arrangements of collagen fibers in the bone matrix, resulting in two microscopic structural organizations. We have a primary bone. The collagen fibers first deposited in osteoid by osteoblasts are randomly arrayed for primary bone. If this material is then mineralized, the result is called a primary or a woven bone. Next, we have a secondary bone. So primary bone is eventually removed entirely or replaced with secondary or lamellar bone, in which the collagen fibers are arranged in layers with all fibers in a parallel arrangement. Woven bone is more quickly assembled, but lamellar bone with its plywood-like structure is actually stronger. We have our elastic fibers as well. Our elastic fibers are found in connective tissues in varying proportions and are most abundant in organs that experience stretching. So, for example, these are our vocal cords, our trachea, and our skin. They are present to some extent in many locations throughout the body. Elastic fibers in loose connective tissue are more dispersed and therefore easier to visualize. Elastin is abundant in a more sheet-like or membrane form in the media of the aorta and other major arteries. We are now going to talk about the functions of the skeletal system. So the skeletal system has 6 important functions. First, they provide support as structural framework. Second, they protect the internal organs such as our brain and heart from injury. 3, they assist body movements in conjunction with muscles. So when muscles contract, they pull bones to produce movement. They also store and release salts of calcium and phosphorus. They also participate in blood cell production or hemopoiesis, and they store triglycerides in adipose cells of yellow marrow as potential chemical energy reserves. We're now going to talk about the structure of bones. So our bones, this is a long bone which consists of a diaphysis, which is our bone shaft. We also have 2 epiphyses, both which are ends of the bone at the joints. And then after that, you have your 2 metaphyses, which are regions between your diaphysis and your epiphysis, right here. Next, we would have, of course, after that, your articular cartilage, which covers both epiphyses. They're right here. And then we have our periosteum. So these are connective tissue that surround the diaphysis. And we have our medullary cavity, which is a hollow space within your diaphysis. And finally, we have your endosteum, which is a thin membrane lining the medullary cavity. Next, we are going to talk about the histology of bones. So our bones contain 4 types of cells. So We have our osteoprogenitor cells, our osteoblasts, our osteocytes, and our osteoclasts. Our osteoprogenitor cells, they are found in inner portion of the periosteum. So our bone stem cells able to differentiate into the other types of cells. And these are our osteoblasts. So these are the bone building cells that secrete matrix. They synthesize and secrete collagen fibers to build ECM, and they do not undergo cell division. Next, we have our osteocytes, which are our mature bone cells. They maintain metabolism, and the same with our osteoblasts, they also do not undergo cell division. Our osteoclasts, they are concentrated in the endosteum. They release lysosomal enzymes and digest proteins and minerals of bone matrix. So this bone breakdown is called bone resorption that is needed for normal repair of bones. So this is involved in the remodeling of bones and causes them to release calcium. So our osteoblasts, so blasts build bones, and our osteoclasts, they carve out bones. So clasts carve out bones. So continuing the histology of bones, let us differentiate the compact bone, which you can see is in the outer layer, and our spongy bone, which is in the inside. Okay, so our compact bone is good at providing protection and support versus our spongy bone, which is lightweight and provides tissue support. Let's now talk about bone formation. So bone formation, also called osteogenesis or ossification, our bones form in 4 situations. So 1, during embryological and fetal development. 2, when bones grow before adulthood. 3, when bones remodel. And 4, when fractures heal. So again, ossification or osteogenesis is the process of bone formation. And ossification takes place in 2 forms, which we will be discussing in the next couple of slides. So bone formation, we have intramembranous ossification. Again, ossification takes place in 2 forms. We have intramembranous and endochondral. Let's discuss intramembranous first. So intramembranous ossification occurs in flat bones when a connective tissue membrane is replaced by bone. So this is the simpler of the 2 methods. So this happens in the flat bones in the skull, our facial bones and mandible, and the medial clavicle. So those bones are formed this way. So these are the steps. So there are 4 processes. So the first one is where cells are still clustered together and they differentiate. So from the osteoprogenitor to osteoblast, the site is the ossification center. So the first step, this is the development of the ossification center. So our osteoblasts will secrete the organic extracellular matrix. After that, so we have the second one where secretion of the extracellular matrix stops and the cells become osteocytes. So calcification, calcium and other mineral salts are deposited and the extracellular matrix starts to harden or calcifies. The third one, so our connective tissues that are associated with blood vessels in the trabeculae will now differentiate into a red bone marrow. And then right here also, if we look at the diagram, so number 3, the formation of trabeculae. So our ECM develops into trabeculae that fuse to form the spongy bone. And the fourth one, so this is the development of the periosteum. So the thin layer of the compact bone replaces the surface of the spongy bone, and the spongy bone will remain in the center. So we're done with the intramembranous ossification. Let's now talk about the endochondral ossification, which replaces cartilage with bone in the developing embryo and fetus. So here we have 6 steps. Number 1, what happens here is there is crowding of cells that forms the chondroblasts. In the second step, the chondroblasts become chondrocytes. So there will be growth by length, which is called your interstitial growth, and then thickness will also happen. So increased thickness will also happen, which is called appositional growth. And at this point, our chondrocytes will hypertrophy and will begin to calcify. With the third step, the pericardium starts to form the bone. And now there is growth of the primary ossification center, where bone tissue replaces most of the cartilage. In our fourth step, the osteoclast breaks some of the spongy bone, leaving a cavity. And then after that, a secondary ossification center develops at the time of birth. And finally, at the sixth step, the hyaline cartilage that cover the epiphysis becomes articular cartilage. Endochondral ossification also occurs in epiphyseal plates of long bones as they grow in length. Now let's talk about, again, we're still at bone formation. We're going to talk about the lengthening of long bones. So the epiphyseal plate is responsible for longitudinal bone growth. This illustration right here on your left shows the zones bordering the epiphyseal plate of the epiphysis. The topmost layer of the epiphysis is the reserve zone. The second zone is your proliferative zone. So this is where your chondrocytes are continually undergoing mitosis. The next zone is the zone of maturation and hypertrophy, where lipids, glycogen, and alkaline phosphatase accumulate, causing the cartilaginous matrix to calcify. So the following zone is the calcified matrix, where the chondrocytes have hardened and die as the matrix around them has calcified. The bottom row is the zone of ossification, which is part of the metaphysis. The newly deposited bone tissue at the top of the zone of ossification is called the primary spongiosa, while the older bone is labeled the secondary spongiosa. So at the zone of ossification, our osteoprogenitor cells invade the matrix and produce osteoblasts that begin creating woven bone on the calcified matrix. Bones continue to grow in length until early adulthood, with the rate of growth controlled by our hormones. Okay, the next slide talks about bone repair. So this one is very important because sometimes a bone is broken and that needs to be repaired. So the steps here are important. So we have, the first one is your hematoma formation, where blood is released from the damaged blood vessels. That forms a hematoma. After that, callus formation happens. So the internal callus forms between the ends of the bones, and the external callus forms a collar around the break. Third, callus ossification happens. So woven spongy bone replaces the internal and external calluses. And then finally, bone remodeling happens. So compact bone replaces woven bone, and part of the internal callus is removed, restoring the medullary cavity. This diagram shows the bone's role in calcium homeostasis. Our bones are the major reservoir for calcium. In here, we have our parathyroid hormone, which is secreted by the parathyroid gland. This increases blood calcium levels. So the parathyroid gland secretes parathyroid hormone when calcium levels drop. Additionally, osteoclasts are stimulated to increase bone resorption, and calcium is released. PTH also stimulates the production of calcitriol by the kidneys to increase calcium absorption in the intestines. We also have calcitonin from the thyroid gland, and this has the potential to decrease calcium levels. Vitamin D enhances the absorption of calcium and phosphate, and thus raises the blood levels of these substances. Alright, now let's talk about aging and its effects on bone tissue. So from birth through adolescence, more bone is produced than is lost during remodeling. In adults, the rates are the same. For older individuals, especially post-menopausal women, they experience a decrease in bone mass when resorption outpaces deposition. Alright, we're done with the connective tissues and the bone and joint. We're now going to talk about the integumentary system. So let's start off with the functions of the integumentary system. So we have protection, cutaneous sensation, temperature regulation, vitamin D production, and excretion. So for protection, the skin protects against abrasion and the harmful effects of ultraviolet light. It also keeps microorganisms from entering the body and prevents dehydration by reducing water loss from the body. The integumentary system also has sensory receptors that can detect heat, cold touch, pressure, and pain. That's why it has functions with sensation. It also functions with temperature regulation, so the amount of blood flow through the skin and the activity of sweat glands help regulate body temperature. It also has functions with vitamin D production, so when exposed to ultraviolet light, the skin produces a molecule that can be transformed into vitamin D, which is an important regulator of calcium homeostasis. And finally, we have excretion, where small amounts of waste products are excreted through the skin and glands. Let's now talk about the structures of the skin. The skin is also referred to as the cutaneous membrane. This covers the external surface of the body, and this is the largest organ of the body in weight. So in adults, the skin covers about 7% of total body weight. The superficial, thinner portion, which is composed of epithelial tissue, is the avascular epidermis. For this reason, if you scratch the epidermis, there is no bleeding. And then we have the deeper, thicker connective tissue portion, which is the dermis. It is vascular, and a cut that penetrates to the dermis will produce bleeding. Deep to the dermis, but this is actually not part of the skin. This is your subcutaneous layer or hypodermis, which consists of areolar and adipose tissues. So the skin and the subcutaneous layer form the integument. Okay, we're starting off with the epidermis first. So this is our epidermis. This receives nutrients and excrete waste products by the diffusion of substances between the epidermis and the capillaries of the dermis. Let's now talk about the types of cells of the epidermis. So the epidermis consists of 4 principal types of cells. We have our keratinocytes, melanocytes, intraepidermal macrophage cells, and tactile epithelial cells. About 90% of epidermal cells are keratinocytes, which are arranged in 4 or 5 layers and produce the protein keratin. So up first is keratin, which helps protect the skin and underlying tissues from abrasions, heat, microbes, and chemicals. And then we have, below that, we have keratinocytes, which release a water-repellent sealant that decreases water entry and water loss and inhibits the passage of foreign materials. Next, we have our melanocytes. Our melanocytes produce melanin, simply put. Melanin is a yellow, red, or brown-black pigment that contributes to skin color and absorbs damaging UV light. So once inside keratinocytes, the melanin granules cluster to form a protective veil over the nucleus on the side toward the skin surface. In this way, they shield the nuclear DNA from damage by UV light. And although their melanin granules effectively protect keratinocytes, melanocytes themselves are particularly susceptible to damage by UV light. And then we have our intraepidermal macrophage, or our Langerhans cells. They participate in immune responses mounted against microbes that invade the skin. Intraepidermal macrophage cells and other cells of the immune system recognize a foreign microbe or substance so that it can be destroyed. Intraepidermal macrophage cells are also easily damaged by UV light. And then we have our Merkel cell. These are tactile epithelial cells and are located in the deepest layer of the epidermis, where they contact the flattened process of a sensory neuron, which is a nerve cell. A structure called a tactile disc, so this is your Merkel disc. Tactile epithelial cells and tactile discs detect touch sensations. The epidermis is composed of 4 layers in thin skin and 5 layers in thick skin. They are from deep to superficial. We're going to start off with stratum basale, which is composed of a single row of cuboidal or columnar keratinocytes, some of which are stem cells that undergo cell division to continually produce new keratinocytes. Next up, we have the stratum spinosum, where the cells in the more superficial layers become somewhat flattened. If you look at this in the microscope, you're going to see that they appear to have been covered with thorn-like spines. That's why it's called the stratum spinosum. So, at each spine-like projection, bundles of keratin intermediate filaments insert into desmosomes, which tightly join the cells to one another. This arrangement provides both strength and flexibility to the skin. Intraepidermal macrophage cells and projections of melanocytes are also present in the stratum spinosum. Now let's talk about the stratum granulosum. The stratum granulosum consists of 3 to 5 layers of flattened keratinocytes that are undergoing apoptosis. Recall that apoptosis is an orderly, genetically programmed cell death in which the nucleus fragments before the cells die. The nuclei and other organelles of these cells begin to degenerate as they have moved farther from their source of nutrition. A distinctive feature of cells in the granulosum is the presence of darkly staining protein granules. This protein, called keratohyalin, is involved in assembling the keratin intermediate filaments into keratin. Also present in the keratinocytes are membrane-enclosed lamellar granules, which fuse with the plasma membrane and release a lipid-rich secretion. This secretion is deposited in the spaces between cells of 3 epidermal layers. You have the stratum granulosum, stratum lucidum, and stratum corneum. Next is your stratum lucidum. This consists of 4 to 6 layers of clear, flat, dead keratinocytes that contain large amounts of keratin and thickened plasma membranes. Finally, we have the stratum corneum, which consists of an average of 25 to 30 layers of flattened dead keratinocytes, but can range in thickness from a few cells in thin skin to 50 or more cells or cell layers in thick skin, and these cells are called your corneocytes or squames. These corneocytes no longer contain nuclei or any internal organelles, and these are arranged like scales or like pieces of a jigsaw puzzle to hold the layers together. This outer stratum of the epidermis, often referred to as the cornified layer, where cells are continuously shed and replaced by cells from the deeper strata. Its multiple layers of dead cells help the stratum corneum to protect deeper layers from injury and microbial invasion. Constant exposure of skin to friction stimulates increased cell production and keratin production, and that results in the formation of a callus, which is an abnormal thickening of the stratum corneum. So again, just to recap from deep to superficial, we have the following layers. You have your stratum basale, and then your stratum spinosum, and then your stratum granulosum, and then your stratum lucidum, and then your stratum corneum. Okay, this slide talks about thick versus thin skin. So we have thin or hairy skin, which covers all body regions except the palms, palmar surfaces of digits, and soles. And then we have our thick or hairless skin, which covers the palms, palmar surfaces of digits, and soles. Next, we have skin pigments under the epidermis. We have melanin, which is produced by melanocytes in the stratum basale. Another common form of melanin is pheomelanin, which is a red-brown polymer of benzothiazine, largely responsible for red hair and freckles. Keratin is a yellow pigment found in plants such as carrots and corn, and we ingest that as a source of vitamin A. It is lipid soluble, and when large amounts of keratin are consumed, the excess accumulates in the stratum corneum and in adipocytes of the dermis and subcutaneous tissue. This causes the skin to develop a yellowish tint, but when you reduce your keratin intake, this yellowish tint also disappears. We have other types of skin pigments like reddening, so blood flowing through the skin imparts a reddish hue, a condition called erythema. And then we have an inflammatory response stimulated by infection, sunburn, allergic reactions, insect bites, or other causes can produce erythema as exposure to the cold and blushing or flushing when angry or hot. We have jaundice, which is due to a buildup of the yellow pigment bilirubin in the skin. This condition gives a yellowish appearance to the skin and the whites of the eyes and usually indicates liver disease. We also have pallor or paleness of the skin, which may occur in conditions such as shock and anemia. This next slide shows two different conditions. We have vitiligo and albinism. Vitiligo is a chronic disorder that causes depigmentation patches in the skin. The precise cause is not known, but is most likely a combination of genetic factors coupled with a disorder of the immune system. So this could be an autoimmune disease. Albinism is a congenital disorder characterized by the complete or partial absence of pigment in the skin, hair, and eyes due to a defect of an enzyme involved in the production of melanin. Now that we're done with the epidermis, we're now going to talk about the dermis, which is the second deeper part of the skin. This is composed of a strong, dense, irregular connective tissue containing collagen and elastic fibers. It is much thicker than the epidermis, and this thickness varies from region to region in the body, reaching its greatest thickness on the palms and soles. Because the dermis is typically thinner in women than in men, many women have the appearance of dimples in the skin referred to as cellulite. The dermis has great tensile strength, which is high resistance to pulling or stretching forces. It also has the ability to stretch and recoil easily. So think about leather that is used for belts, shoes, baseball gloves, and basketball. So this is actually dried and treated animal dermis. As is typical of all general connective tissue, the cells present in the dermis are scattered and include fixed cells and wandering cells. The predominant fixed cells are fibroblasts. The wandering cells include your macrophages, mast cells, eosinophils, neutrophils, and dermal interstitial dendritic cells, which are immune surveillance cells. Blood vessels and nerves, along with glands and hair follicles, are embedded in the dermal layer. The dermis is essential to the survival of the epidermis, and these adjacent layers form many important structural and functional relations. Based on its tissue structure, the dermis can be divided by an indistinct boundary into a thin, superficial papillary region and a thick, deeper reticular region, which we will further talk about in the next slides. So let's talk about the papillary region, which makes up about 1/5 of the thickness of the total layer. So the dermal papillae that you can find in the papillary region can vary greatly in size and number throughout the different parts of the dermis. They are taller and more numerous in sensitive regions of skin that experience more mechanical stress. In thin skin covering most of the body, the dermal papillae are relatively few in number, small, and irregularly scattered. The papillary layer also contains blood vessels that supply the overlying epidermis with oxygen and nutrients. They remove waste products and aid in regulating body temperature. The ridges, as mentioned in this slide, increase friction and improve the grip of the hands and feet. Next, we have the reticular region of the dermis, which is attached to the subcutaneous layer, contains bundles of thick collagen fibers, scattered fibroblasts, various wandering cells, and some coarse elastic fibers. The collagen fibers in the reticular region are arranged in a net-like manner and in a more regular formation than those in the papillary region. The more regular orientation of the large collagen fibers aligns with the local tensile forces to help the skin resist stretching. In certain regions of the body, collagen fibers within the reticular region of the dermis tend to orient more in one direction than another because of natural tension resulting from bony projections, orientation of muscles, and movements at joints. Now let's talk about tension lines or lines of cleavage in the skin, which indicate the predominant direction of underlying collagen fibers. Knowledge of tension lines is especially important to plastic surgeons. For example, a surgical incision running parallel to the collagen fibers will heal with only a fine scar. A surgical incision made across the rows of fibers will disrupt the collagen, and the wound tends to gape open and heal in a broad, thick scar. The combination of collagen and elastic fibers in the reticular region provides the skin with strength, extensibility, the ability to stretch, and elasticity, the ability to return to original shape after stretching. The extensibility of skin can be seen readily around joints and in pregnancy and obesity. Let's now talk about ridges. The surfaces of the palms, fingers, soles, and toes are marked by a series of ridges and grooves. These epidermal ridges serve multiple functions. One, they increase the surface area of the epidermis and thus function to increase the grip of the hand or foot by increasing friction. And because there is increased surface area, this also increases the number of corpuscles of touch and thus increases tactile sensitivity. Additionally, the interdigitating pattern between epidermal ridges and dermal papillae creates a stronger bond between the epidermis and dermis in regions of high mechanical stress. Because the ducts of sweat glands open on the tops of the epidermal ridges as sweat pores, the sweat and ridges form the fingerprints or footprints when a smooth object is touched. The epidermal ridge pattern is in part genetically determined, but even identical twins have different patterns. So let's talk a bit about tattoos. This is something that's just good to know. Again, tattoos are created by injecting ink with a needle that punctures the epidermis and moves between 50 and 3,000 times per minute as the ink is deposited in the dermis. However, they can fade over time due to exposure to sunlight, improper healing, picking scabs, and flushing away of ink particles by the lymphatic system. Tattoos can be removed by lasers in a series of treatments that use concentrated beams of light. In the procedure, tattoo inks and pigments selectively absorb the high-intensity laser light without destroying normal surrounding skin tissue. The laser causes the tattoo to dissolve into small ink particles that are eventually removed by the immune system. So, our skin also contains different types of sensory receptors found in different layers. You have your superficial layer and your deep layer. Like some areas of the dermis, the subcutaneous layer also contains encapsulated nerve endings called lamellated Pacinian corpuscles that are sensitive to pressure. Now that I've mentioned the subcutaneous layer, let's talk about it a little bit more. So this is deep to the dermis, and this is also referred to as the hypodermis. This connective tissue layer, which is not part of the skin, differs from region to region in the body. In some areas, it is a thin layer comprised of the loose connective tissue called areolar tissue, while in other regions, it is a thick, tough layer of fibrous bands of collagen accompanied by adipose tissue. The subcutaneous layer has multiple functions. It functions as a loose binding tissue that unites the upper layers of the skin to deeper structures, while at the same time allowing the skin to move freely over these deeper structures. In certain regions, such as the skin of the soles and palms, the subcutaneous layer forms tough, fat pads composed of fibrous bands of collagen and adipose tissue. This absorbs shock and protects underlying muscle and bone. As noted previously, the subcutaneous layer is also the principal site of energy storage in the body. Adipose tissue is an active metabolic tissue with numerous nerve endings and rich vascular networks that help regulate and mobilize the energy stores. Finally, the subcutaneous layer serves as a layer of insulation that helps retard heat loss from the body. Let's talk a bit about the accessory structures of the skin. So we have the hair and hair follicles, which are present on most surfaces except the palms, the anterior surfaces of fingers, and the soles of the feet. These are composed of dead keratinized epidermal cells, and genetics will determine the thickness and distribution. So as you can see, it is affected by physiological or emotional stress, and this event is called your goosebumps or your goose flesh. We're now going to talk about the glands of the skin. So we have 4 types of glands. We have the sebaceous glands, eccrine sweat glands, apocrine sweat glands, and our ceruminous glands. We'll first talk about our sebaceous glands or oil glands, which are connected to hair follicles, and they help prevent water loss and inhibit bacterial growth on the surface of the skin. So our sebaceous glands vary in size and shape and are found in the skin over all regions of the body except the palms and soles. Because of their relationship to the hairs, sebaceous glands are most numerous where hairs are most numerous. So they would be small in most areas of the trunk and limbs, but large in the skin of the breast, the face, the neck, and the upper chest. The sac-like base of a sebaceous gland has a lining of cuboidal cells that resemble basal epithelial cells. And a lot of cells, or these multiple generations of cells, will pile on top of each other, and the lumen will gradually fill with cells. And these cells differentiate by developing large lipid vesicles in their cytoplasm, and they will become so distended and ruptured, and they fill the duct of the gland with an oily secretion called sebum, which is derived from released lipid-filled vesicles and cellular debris. Sebum is a mixture of fats, cholesterol, proteins, and inorganic salts, and they prevent excessive evaporation of water from the skin. And then we have your eccrine sweat glands, which are also called your merocrine sweat glands. They are simple coiled tubular glands that are much more common than apocrine sweat glands. So they are distributed throughout the skin of most parts of the body except for the margins of the lips, the nail beds of the fingers and toes, the glans penis, the glans clitoris, the labia minora, and eardrums. Our eccrine sweat glands are most numerous in the skin of the forehead, palms, and soles. The sweat produced by eccrine sweat glands consists primarily of water with small amounts of ions, mostly sodium and chloride, etc. So as sweat evaporates, large quantities of heat energy leave the body surface. The homeostatic regulation of the body temperature is known as thermoregulation. And the main role of eccrine sweat glands is to help the body to achieve thermoregulation, which is known as thermoregulatory sweating. During the thermoregulatory sweating, sweat first forms on the forehead and scalp, and then extends to the rest of the body, forming last on the palms and soles. Sweat that evaporates from the skin before it is perceived as moisture is termed insensible perspiration. Sweat that is excreted in larger amounts and is seen as moisture on the skin is called sensible perspiration. Eccrine sweat glands also release sweat in response to an emotional stress, such as fear or embarrassment. This type of sweating is referred to as emotional sweating or a cold sweat. Emotional sweating first occurs on the palms, the soles, and axilla, and then spreads to the other areas of the body. Next, we have the apocrine sweat glands, which are located mainly in hairy skin. Apocrine sweat glands, like eccrine sweat glands, are simple, coiled tubular glands, but they have larger ducts and lumens than the eccrine sweat glands. They are found mainly in the skin of the axilla, the groin, the areola of the breasts, and bearded regions of the face in adult males. Their secretory product is slightly viscous compared to eccrine secretions and contain the same components as eccrine sweat plus lipids and proteins. So at first, this substance will start off as odorless, but as it spreads onto the hairs, the bacteria will decompose the proteins, and this will create a strong musky odor that is called body odor. In women, cells of apocrine sweat glands enlarge at about the time of ovulation and shrink during menstruation. Eccrine sweat glands start to function soon after birth, but apocrine sweat glands do not begin to function until puberty. Apocrine sweat glands are stimulated during emotional stress and sexual excitement, so these secretions are commonly known as a cold sweat. In contrast to eccrine sweat glands, apocrine sweat glands are not active during thermoregulatory sweating. Finally, we have ceruminous glands, which are modified sweat glands. So the combination of the secretion of ceruminous and sebaceous glands will be called cerumen or earwax, and cerumen will also waterproof the canal and will keep bacteria and fungi from entering. Just a quick recap of everything we've talked about. So we talked about the different functions of the integumentary system, its roles in protection, cutaneous sensation, temperature regulation, vitamin D production, and excretion. So for protection, we've talked about keratin, which protects underlying tissues from microbes, keratinocytes that resist invasion by microbes. We have lipids that inhibit evaporation of water from the skin surface, thus protecting against dehydration. We've also discussed about oily sebum from the sebaceous glands that protect the skin and hairs from drying out, which also contains substances that kill bacteria. We also discussed about the pigment melanin that helps shield against the damaging effects of ultraviolet light. Also, we've talked about the intraepidermal macrophage cells that alert the immune system to the presence of potentially harmful microbial invaders. We also have macrophages in the dermis that phagocytose bacteria and viruses. So we have roles in cutaneous sensation that, there is a variety of nerve endings and receptors distributed throughout the skin. So these are the tactile discs of the epidermis, the corpuscles of touch in the dermis, and hair root plexuses around each hair follicle. It also has roles in thermoregulation, so the skin contributes to homeostatic regulation of body temperature in two ways. So by liberating sweat at its surface and by adjusting the flow of blood in the dermis. For synthesis of vitamin D, this requires activation of a precursor molecule in the skin by UV light or ultraviolet rays in sunlight. And then enzymes in the liver and kidneys then modify the activated molecule, finally producing calcitriol, which is the most active form of vitamin D. So calcitriol is a hormone that aids in the absorption of calcium from foods in the GI tract into the blood. Vitamin D is believed to enhance phagocytic activity and increase the production of antimicrobial substances by phagocytes. It also regulates immune functions and helps reduce inflammation. Finally, the skin has roles in excretion. So besides removing water and heat from the body, sweat also is the vehicle for excretion of small amounts of salts, carbon dioxide, and two organic molecules that result from the breakdown of proteins, which are ammonia and urea. So our skin physiology also consists of vascular supply, nerve supply, and skin nutrition. The dermis contains several microvascular blood vessel plexuses and lymphatic vessels that are parallel to the skin surface. The larger arterioles and venules are in the deep reticular layer, with smaller vessels extending into the papillary layer and terminating in capillary loops. For the nerve supply, because of its large and superficial surface area, again, the skin contains the sensory receptors necessary for the body to process the external environment. The nerve endings are either unencapsulated or encapsulated. We're now going to talk about wound healing. So there are two types of wound healing. We have epidermal wound healing, which starts off with division of the stratum basale and migration across the wound and thickening of the epidermis. Next, we have your dermal wound healing, which has the inflammatory phase and then the maturation phase. Our skin is also affected by aging. There are changes in the integumentary system with age, and these are all listed here. As shown in the previous slide, with age, there is an increased susceptibility to pressure ulcers. Now let's talk a bit about pressure ulcers. There are stages or different levels depending on the severity of the involved layer of the skin. So we have stage 1, where this is just erythema of the skin. Stage 2, this is erythema with the loss of partial thickness of the skin, including the epidermis and part of the superficial dermis. Stage 3, this is a full thickness ulcer that might involve the subcutaneous fat. Stage 4 is a full thickness ulcer with the involvement of muscle or bone. We also have an unstageable pressure injury, which is obscured full thickness skin and tissue loss. Full thickness skin and tissue loss in which the extent of tissue damage within the ulcer cannot be confirmed because it is obscured by slough or eschar. Skin cancer can also affect the integumentary system, and there are 3 major types as seen in the slide. So you have your basal cell, squamous cell, and malignant melanoma. Let's discuss burns. It's important to talk about burns because as physical therapists, we are involved in post-burn rehabilitation for clients with burns, and we assist them in regaining endurance, strength, and balance that are needed for activities of daily living. So it's important that we know how burns happen and to some extent the different levels or the different degrees of the burns. So we have the first degree burn, which involves only the epidermis. It is characterized by mild pain and redness but no blisters. Generally, a first degree burn will heal in about 3 to 6 days and may be accompanied by flaking or peeling. An example of a first degree burn is a mild sunburn. And then we have a second degree burn that destroys a portion of the epidermis and possibly parts of the dermis. Associated structures such as hair follicles, sebaceous glands, and sweat glands usually are not injured. So if there is no infection, second-degree burns usually heal without skin grafting in about 3 to 4 weeks, but scarring may result. First and second-degree burns are collectively referred to as partial thickness burns. And then we have a third-degree burn or full-thickness burn, which destroys a portion of the epidermis, the underlying dermis, and associated structures. With full-thickness burns, there is marked edema and the burned region is numb because sensory nerve endings have been destroyed. Skin grafting may also be required to promote healing and to minimize scarring. This is the Rule of Nines, also known as the Wallace Rule of Nines. This is a tool used by trauma and emergency medicine providers to assess the total body surface area involved in burn patients. This tool is only utilized for second-degree and third-degree burns, which is also referred to as your partial thickness and your full thickness burns. So if we look at the picture right here, we count 9% if both the anterior and posterior surfaces of the head and neck are affected. So that's 4.5 anteriorly, 4.5 posteriorly. We count 9% for both the anterior and posterior surfaces of each upper limb. So that's 4.5 anterior, 4.5 posterior. That's 9% per upper limb, total of 18% for both upper limbs. We count 4 times 9 or 36% for both the anterior and posterior surfaces of each or of the trunk, including the buttocks. We count 9% for the anterior and 9% for the posterior surfaces of each lower limb. That's 18% per lower leg, and that's a total of 36% for both lower limbs. And we count 1% for the perineum. Okay, so let's have an example here. We have a 58-year-old female patient that has superficial partial thickness burns to the anterior head and neck, front and back of the left arm, front of the right arm, the posterior trunk, front and back of the right leg, and back of the left leg. Using the rule of nines, calculate the total body surface area percentage that is burned. So if we are going to shade the different parts mentioned for this partial thickness burns, we have the anterior head and neck, we have the front and back of the left arm, we have the front of the right arm, we have the posterior trunk, and we have the front and back of the right leg, and then we have the back of the left leg. So we put the values for each of those shaded parts, and when we add all of those, we result to a total of 63% total body surface area. So that is how we use the rule of 9. So that's how the diagram will look like or will be presented on, let's say, charts that you receive, for example. Okay, for the last slide, this table just shows the difference between the rule of nines of adults versus children. If you're wondering why are they different, it's because the anatomy or the size of children are very much different from adults, so we become more proportionate as we grow older. If you remember, when you're a kid, your head is significantly bigger than your body. That's why if you look at the table, about 18% is already dedicated for the portion of the head. Alright, that's it for this lesson. If there is anything you didn't understand, make sure to take note of them. Thank you very much for your time, and these are my references.